Hello boys, welcome back to another session of online biology video tutorials, and this session is meant for 5th July 2021, and this is for class 12th biology students. As all of us know, we have been discussing about sexual reproduction in flowering plants, that is chapter number 2. In our previous sessions, we were talking about the concept of double fertilization. I said double fertilization is very important not only for the sake of board exams, even for the general life purpose. Because of double fertilization, we get a lot of new plants, new embryos, and then the endosperm that is present because of triple fusion will help the seeds to have that. So from the embryo, from the zygote, the fruit will develop. We said developed ovary is a fruit, and inside when we cut the fruit there will be seed. So that seed is technically developed ovule. So we say seed is the fertilized ovule formed inside fruits. It is the final product of sexual reproduction. It consists of seed coats. That seed coat is cotyledons and embryo axis, as you are seeing here, where they, depending upon how many cotyledons are there, the seeds are of two types, or in general plants are of two types, dicotyledonous and monocotyledonous. As the name suggests, di means two coats will be there, two cotyledons. See here, cotyledon 1, cotyledon 2. So since two cotyledons are there, dicots, and here you can see the presence of only one cotyledon, so monocot seed. So in dicotyledonous, as you are seeing, there will be outside externally seed coat, then there is cotyledons coverings, and then there is a presence of hypocotyle and epicotyle structures which lead to germination of new plant. And similarly, in case of monocot seed, you can see the seed coat, then presence of one cotyledon inside, and this is what we said, the triple fusion product, endosperm, which is the nutritious tissue. And you have epicotyle, which leads to formation of stem, hypocotyle, which leads to formation of radicle, and radicle becomes a future root. So the cotyledons are simple, generally thick and swollen due to storage of food, as in legumes. So we can see the vertical section of the seeds, both monocot and dicot. And mature seeds are of two types. On maturity we can see this, non-albuminous seeds and albuminous seeds. What are non-albuminous? As the name suggests, there is no residual endosperm, there is no albumin. So without albumin, the seeds, if they are present, they are non-albuminous seeds, as it is completely consumed during embryonic development. As the embryo develops, the developing embryo consumes the total endosperm that is available. So the seed will be left out without any endosperm. So such seeds are called non-albuminous seeds. Example, the groundnut and beans. You can see the bean seed here. There is no, you can see there is no presence of endosperm. Completely it is consumed. So epicotyle, hypocotyle, radicle. Radicle is nothing but future root. Then seed coverings, cotyledons, and outer coat, seed coat. Similarly, watane, beans, peas. You can see here also plumule, all that, and then cotyledons, of course, with storage. And then in corn seed also you can see here, there is a presence of what we call the cotyledon, then radicle, hypocotyle, and look at this portion, endosperm. So seeds where endosperm is present like this, they are called albuminous seeds. So they retain some endosperm. Example, wheat, maize, barley, castor seeds, coconut, and sunflower. All these will have the endosperm. So they are called albuminous seeds. And non-albuminous seeds, they have no residual endosperm. as it is completely consumed during embryonic development. Example we have seen pea, groundnut and beans. Albuminous, they retain some endosperm, wheat, maize, barley, castor, coconut and sunflower. Now what is important is here the vertical section. Many times this is asked in the exam. So the vertical section of the maize. So you need to practice this diagram wherein you have got the pericarp, outer coat, then endosperm. See, majority portion is endosperm, and then you have got the scutellum, majority portion again. Then coleoptile, plumule, radicle, and coleorhiza. Similarly, this diagram normally it is asked for say 2 marks or 3 marks. You need to practice this albuminous seed. Similarly, the vertical section of hypocotyle one, that is the seed coat, endosperm, cotyledons. So sometimes this is also asked as a 1 mark question. So you need to practice these diagrams. Occasionally in some seeds like black pepper, beet, remnants of nucellus are also present. It is called perisperm. So if nucellus, which is a nutritive tissue, remember, if that is also present in the form of perisperm, so that perisperm is nothing but remains of nucellus. So here also you can see the embryo getting developed and large amount of food storage and then seed coat. Similarly in peanut seeds, that is groundnut, you can see the presence of enormous amount of this embryonic seed with leaves, and then the development of embryo. Plumule is going to be future stem, radicle is going to be future root. And then of course 2 cotyledons, cotyledon 1, cotyledon 2, dicot. So integuments of ovules harden as tough protective seed coats. It has a small pore that's called micropyle through which oxygen and water enter the seed during germination. As the seed matures, it becomes dry by reducing water content to 10 to 15% of moisture by moss. So we are seeing every day the seeds are getting dried. The metabolic activity of the embryo slows down. It may enter a state of inactivity, we say dormancy, seed dormancy. So the period of seed dormancy also varies from seed to seed. Under favorable conditions, like good moisture content is there, oxygen is available, temperature is suitable, then the seeds will germinate, as you are seeing here. Seed germination you can see. And then the advantages of seeds, since pollination and fertilization are independent of water, seed formation is more dependable. Better adaptive strategies for dispersal to new habitats. It helps the species to colonize in other areas. They have food reserves, so young seedlings are nourished until they are capable of photosynthesis. So seed dispersal will take place. The hard seed coat protects the young embryo, as you are seeing the cotyledons. Being products of sexual reproduction, they generate new genetic combinations and variations. So seeds will lead us to variations. Dehydration and dormancy of mature seeds are crucial for storage of seeds. It can be used as food throughout the year and also to raise crop in next session. We are using so many seeds as our food, and we are also using the seeds for the next session for the crop. So in few species, the seeds lose viability within few months. Seeds of many species live for several years, as I said, because of seed dormancy. Some seeds can remain alive for hundreds of years. The oldest is that of lupine, Lupinus arcticus. You can see here, Lupinus arcticus. So hundreds of years. It is excavated from Arctic tundra. The seed germinated and flowered after an estimated record of, look at that, 10,000 years of dormancy. 10,000 years, my goodness. 2,000 years old viable seed is of the date palm. Okay, Phoenix dactylifera, here, date palm. Discovered during the archaeological excavation at King Herod's palace near the Dead Sea. So that is the viability. The next, the ovary develops into a fruit, as we have been talking about. Transformation of ovules into seeds and ovary into fruit proceeds simultaneously. The wall of ovary develops into pericarp, as you are seeing here. The pericarp consisting of exocarp, mesocarp, endocarp, and this forms the fruit wall. And the fruits may be fleshy, like in guava, orange, mango, or it may be dry, like groundnut and mustard. So you can see here groundnut and mustard. The fruits are of two types. True fruits. What are true fruits? Fruit that develops only from ovary. So if ovary develops into a fruit, then we say it is true fruit. Other floral parts degenerate and fall off. Example, most of the plants will give fruits in this way. But there are false fruits. In this, the thalamus also contributes to fruit formation. Like in apple, you can see here, apple, strawberry, okay, strawberry, cashew nut. In all this, the fruit is false fruit. We say pseudo fruit because it is not only ovary. Ovary plus thalamus becomes fruit. That's why we call apple, strawberry, cashew nut as false fruits. In some species, fruits develop without fertilization. Look at that. Normally, fertilization must, but such fruits which are developed without fertilization, this is important. They are called parthenocarpic fruits. Example, banana. So, banana is a parthenocarpic fruit. Why is it so? It develops without fertilization. So, parthenocarpy can be induced through the application of growth hormones. Such fruits are seedless. So nowadays Bijapur also is famous for seedless grapes, seedless pomegranate, and all this is because of spraying of growth hormones. And this process is called parthenocarpy, development of seedless fruits without fertilization. And then there is something called apomixis. This is the production of seeds without fertilization. Now here see, there we said fruit, parthenocarpy. Apomixis, seeds. You must remember this. So apomixis is the production of seeds without fertilization. Example, some species of Asteraceae, you can see here, Asteraceae and grasses. It is a form of asexual reproduction that mimics sexual reproduction. That is the significance of apomixis, development of seeds without fertilization. And then in some species, diploid egg cell is formed without reduction division and develops into an embryo without fertilization. This is the secret. How apomixic seeds are formed. This is the thing. There is no meiosis in the diploid egg cell. In many species, like citrus, mango varieties, some nucellular cells surrounding the embryo sac divide, protrude into embryo sac to form embryos. Thus, each ovule contains many embryos. So, occurrence of more than one embryo in a seed is called polyembryony. You can see here polyembryony. It is the integument, okay, nucellus, nutritive tissue, normal embryo, this one, you are seeing normal embryo, and then nucellular embryo, see 1, 2, 3, 4. So since more than one embryo is formed, this is called polyembryony. Okay. Then importance of apomixis. If the seeds from hybrids are sown, the plants in the progeny will segregate and lose hybrid character. This is the significance. Production of hybrid seeds is costly, so hybrid seeds are also expensive. If the hybrids are made into apomix, there is no segregation of characters in hybrid progeny. So farmers can keep on using hybrid seeds to raise new crop. This is the importance of apomixic seeds. As we are seeing here, this is how we raise the apomixic seeds. And this is the most crucial part of the entire chapter. The purpose of knowing this chapter is to not only go with the conventional sexual reproduction through which we get the developed fruit, that is developed ovary into a fruit, developed seed into a developed ovule into a seed, but we can also focus on the polyembryony and apomixis. So thus we can help the farmers. So keep on studying, draw the diagrams, practice the diagrams, and I said in this chapter the most important concept is double fertilization. Apomixis, there will be a short notes. Polyembryony, there will be short notes. Parthenocarpy, there will be short notes. Then the microsporogenesis is a short notes.